Welcome. Today we're going to be taking a look at the nervous system. This is going to be a break from the Blosson book today. And I'm going to start out speaking just a little bit about the special senses and the senses. And then I'm going to use the rest of our time to basically go back and sort of rehash, resynthesize, unpack some of the information from the previous discussions on the nervous system. And in large part, this will serve as an overview of much of the material that is involved on exam 3. So when you think of the senses, you probably are thinking back to kindergarten, first grade, when you first learned about the senses. And we were probably taught that there are 5 senses, and they're shown here on the screen. Basically, that we have the sense of touch involving the skin. We have the sense of smell with the nose. We have the sense of hearing with the ear. We have vision with the eye. And we have taste with the tongue. And that is all correct, of course, but we also want to go back and make sure we don't forget about balance. Balance is another important sense that is largely facilitated by our inner ear, and you'll be listening to a larger presentation on the ear, specifically on balance, at the beginning of the material for the next exam. So you'll learn a lot more about balance. As I go through these senses, I hope that one of the things that pops up in your head are the cranial nerves that are involved with these. So if we think about touch, touch is really what's considered a general sense. It is something that is from head to toe, that you have the ability to feel and touch on the surface of your skin. The other senses are considered the special senses. So those are smell, or called olfaction. There's hearing or audition. There's balance, or the vestibular function. There's vision, and then there is taste or gustation. As I go through these, though, I want you to be thinking about the cranial nerves that are involved. You've been introduced to the cranial nerves, and I'll be bringing those up quite a bit in this test. So let's just for a moment think about what cranial nerves are involved with each of these senses. So if I say taste, recall that there are 2 cranial nerves that are carrying that taste sensation from the receptors in the tongue to the brain. The front 2/3 of the tongue, that is facilitated by cranial nerve number 7, the facial nerve, and the back 2/3 of the tongue, those taste buds send their signals through the glossopharyngeal nerve number 9. For vision, you know that the optic nerve, cranial nerve number 2, is responsible for sending information from the back of the eye, the retina, through the optic nerve and to the brain. For the ear, both the hearing and vestibular functions, that information is sent through cranial nerve number 8, the vestibular cochlear nerve. For smell, you know that I'm pointing here to the olfactory nerve. This is cranial nerve number 1. And for the skin, the only cranial nerve that's involved here is involving the sensations on the face. And on the face, recall that cranial nerve 5, the trigeminal nerve, had 3 regions: the upper ophthalmic, the upper jaw, and the lower jaw, and each of those regions was responsible for feeling on your face. So just on your face and on your head, that is cranial nerve number 5, the trigeminal nerve. The rest of your body is the sensation of touch is dealt with spinal nerves, from C2 all the way down through the sacral nerves, and those are the dermatome regions that I'll discuss a little bit more detail with you in a little bit. So in lab, you have been introduced to the anatomy of the eye and the anatomy of the ear, and that's really the anatomy that I want you to know regarding the senses. We're not going to go into any specific details about the taste bud structures that are involved with taste, nor anything special about smell or the other senses, but I do want you to specifically know the structures of the eye and the structures of the ear. So the next 3 slides are just going to simply go through that. This should be a review from lab. So when we think about the eye, you know that there are 3 layers, 3 tunics, if you will. And start here. So the white of the eye, that is the sclera. That is the sclera. And then the middle layer is the choroid, right? The choroid layer. And that's where all the blood vessels are. And then the inner layer that we find mostly just in the posterior portion, this is where the retina is located. So 3 main layers of the eye. And other important structures are as follows. If you were to touch the front of your eye, that is the cornea. Now the cornea is an avascular structure, but it's highly innervated. If you've ever scratched your cornea, you know that's a very painful scenario. But there's no blood vessels in this clear layer, and that makes sense because otherwise you'd be seeing blood vessels as light traveled through that thin layer and that information was brought into the eye. The iris is simply the colored muscle of your eye. Remember, iris means rainbow. So this is the colored muscle. And as the iris opens and closes, constricts and dilates, it makes the pupil larger or smaller. So the pupil is simply the opening through which light passes. As light comes into the eye, there is going to be a water-filled area, a fluid-filled area here in the very front. In the lab, you saw this filled with aqueous humor. That's basically the liquid that maybe came out or was left on your dissection tray as you cut into the eye. As the light comes in, goes through the aqueous humor, it's going to hit the lens. The lens would be a perfectly clear structure in a living donor, unless there's some disease to it. In your dissections, you saw a cloudier looking lens simply from the fixative or the preservatives. Behind the lens, you have a large empty space, not empty, but filled with vitreous humor. That's the jelly that you saw when you did your dissection. And then as light comes through, hits the lens, it gets redirected back to the retina. And the retina is the layer that has all of the nerve endings that receive that light. That information is then sent through the optic nerve. We see that here. You saw that on the back of your eye as well. That is otherwise known as cranial nerve number 2. This particular drawing does give us 2 muscles to look at. Remember, there are 6 muscles around the eye. You learned those back in the muscle lab. But the top would have the superior rectus, and we also see here a little bit of the inferior rectus. Recall that the superior and inferior rectus muscles are controlled by the 3rd cranial nerve, the ocular motor nerve. Looking at this directly, if we look at the eye straight on, again, now it's more clear to see the pupil, just the opening, the iris, the colored muscle around the eye, and the sclera is the white of the eye, and that sclera continues all around the eye. There are no additional structures listed here that I didn't already mention, except here the aqueous humor. That liquid is labeled being here in what's called the anterior cavity. And everything else is as I already described. So just know the structures of the eye and also know the structures of the ear. This was also introduced to you in the nervous system lab. Recall that the ear is broken up into 3 regions: the outer ear, sometimes called the external ear, the middle ear, and the inner ear, sometimes called the internal ear. And structurally, we have the pinna. The pinna is also called the auricle, the flappy portion of the ear. It's mostly cartilage. And then there's the opening. This is the ear canal. This is any place where a Q-tip could reach in. Remember that the ear canal is passing through the external auditory meatus. that is the tube-like passageway through the temporal bone. The outer ear goes as far as the eardrum. You're going to refer to the eardrum as the tympanic membrane. Once you get past that tympanic membrane, you are now in the middle ear. This is normally an air-filled chamber. There are 3 important bones in here. And recall that back in the skeletal lab, we learned all of the bones of the body except for these ossicles. So now it's the time to learn and name them. So from lateral to medial, we have the malleolus, the malleus, the incus, and the stapes. You'll also hear stapes. So the malleus, the incus, and the stapes. These 3 bones, again, are collectively called the ossicles. Also found within the middle ear is the opening to the Eustachian tube. You've seen 3 names for this tube. This is also called the auditory tube, and it's referred to as the pharyngeal tympanic tube because it goes from the pharynx up to the tympanic membrane. The stapes is sitting on an oval window. It's not labeled here, but the oval window is what the stapes is sitting on. Here is the round window that I'm pointing to now. Once you go through, or once those sound waves pass through the oval window, you're now in the vestibule, the opening of the inner ear. As you look off to one side, you see the 3 semicircular canals. As you look off to the other side, you would see the cochlea. The cochlea is the center for hearing. Within the cochlea, there are hair cells, basically nerve endings, that are responding to both the height of the sound waves. The height refers to the amplitude or the loudness of the sound, and also to the frequency of those sound waves. The frequency is how the pitch is. Is it a low pitch or a high pitch? And the hair cells within the cochlea interpret all of that information, and that information is sent through the auditory branch of the vestibular cochlear nerve. This is called the auditory branch or the cochlear branch of the vestibular cochlear nerve. On the other side, within this vestibule region, there's 2 other areas. You saw them in lab, the utricle and the saccule, as well as these 3 semicircular canals. Also, over here, there is fluid and there are hair cells. But this region of the ear is responding to head movement and to acceleration of the head. And as a result, that fluid pushes and bends on those hair cells, and that information is perceived by your brain as movement. And that helps to coordinate your balance as well as your vision. Again, we'll learn more about the vestibular system in a special set of presentations coming up, not on this exam, but will be presented right after exam 3, and that will be part of your exam for information. So that vestibular information is going to travel through the vestibular branch of the 8th cranial nerve. So the vestibular branch and the auditory branch combine to make the 8th cranial nerve. So that's really what I want you to know for the ear. The rest of this presentation, I want to basically almost treat this like a review for the exam. And it may not feel like a review. It may feel like you're learning some new material, and you are, but you're also sort of synthesizing much of what has already been introduced to you. So these slides are posted for you to follow along. I would suggest that you print them out and put notes on them, or at least be very mindful of this information. So the nervous system, as I described in the first presentation, can be divided up into a couple different ways. We can take a look at the sensory division, and you can take a look at the motor division. Now, this is more of a functional organization. I also described to you how the nervous system can be broken up into the peripheral and central nervous system. Remember, the central nervous system is just the brain and the spinal cord, and the peripheral nervous system would be all of those structures outside of the brain or the spinal cord. That's an anatomical division. This is a functional organization. So we have the sensory, also called the afferent division. Now, I will normally call it afferent just to overemphasize this, but you know from vocab that AF means toward and fare means to carry. So this is the division that is carrying toward the central nervous system. This is the information from the environment, either your external environment or your internal environment. And this is the information that's going toward the central nervous system, sending that information to the spinal cord and then up to the brain. And then there's the motor side, or the efferent side. Remember, EF means away from. So the efferent is moving away from the central nervous system back toward the rest of the body. The sensory division can be further divided into the somatic sensory. This simply means the information coming from essentially the surface of your body, that is, from your skin, from the connective tissue down around your muscles. Your joint structures are sending information. Right now, if you are sitting, your knees are probably around 90 degrees. And if you close your eyes, you are aware of that. You're aware of your body's position. You're aware that your knees are about 90%. You can lift your arm up and flex your arm, and you know that your elbow is somewhere around 90 degrees or that your arm is straight out. So you are aware of your joint structures because of this somatic sensory information. Your muscles are also sending information to your nervous system through this branch, through the somatic sensory side of things. And your special senses, so vision, hearing, taste, smell, balance, all of those through those cranial nerves are also somatic sensory. In addition, you have visceral sensory. Visceral sensory, you know, visceral means your internal organs. So this is the information coming in from your viscera. You are aware of a full bladder. You are aware of a full stomach, or maybe an empty stomach. You are aware that you're breathing heavily or that your heart is beating fast or slow. That information is being sent through this side of your nervous system, through the visceral sensory side of things. We talked about how the vagus nerve, right, sends signals up from the gut and from the bladder and from the heart. Then on the motor side, you have the somatic motor. The somatic motor side is really your voluntary nervous system, and you know that the only muscles that you have voluntary control over are your skeletal muscles. All the muscles we named in lab, 4 and 5, are voluntary skeletal muscles. So those muscles are controlled through these motor neurons, what we call the somatic motor nervous system. Then there is the autonomic motor side, and this is where you have all of your involuntary nervous system controls. So think cardiac muscle, smooth muscle, and glandular secretions. So these, you know, are all involuntary responses. Let's take a look and put all of this together. So in this diagram, we have, looks like the biceps brachii, right? And let's say that we have the biceps brachii, we have the spinal cord over here, and we have, it looks like just a part of the shoulder. So something is tickling, let's just describe, something is tickling her shoulder. Let's say there's a fly. So a fly has landed on her shoulder, and there are nerve endings, receptors, that pick up that signal. That signal is going to be sent through a sensory neuron, right? Afferent sensory neuron. And you know that that signal is going to go into the posterior root of the spinal nerve. Here we see the cell body for that sensory neuron. Recall that the sensory neurons are unipolar. And if we could zoom in even closer, we might see satellite cells around that sensory cell body neuron, because remember, those satellite cells are glial cells of the PNS. So we have the unipolar cell body for the sensory neuron, and we see that it comes into the spinal cord, synapses with an interneuron. The interneuron is shown here in purple. Remember, interneurons are neurons that are only contained completely and wholly within the CNS. So we're in the spinal cord here. That interneuron, what we're not seeing here, to be completely clear, is that this interneuron would then be sending signals up the spinal cord, up through ascending tracts, up to the brain, specifically to the part of the brain that receives sensory information, and that would be the parietal lobe. So this information is going to go up to the sensory strip of the of the brain, of the parietal lobe, and then signals are going to go from the parietal lobe, from the sensory strip, over to the motor strip. Remember, the motor strip is in the frontal lobe. Frontal lobe is going to send those instructions back down. The other thing that this picture is not showing yet is that this is all going to decussate. Recall that decussation is the crossing over. 90+% of all of your neurons cross over. So here we're seeing one side of the spinal cord, and it would cross over to the other side on its way to and from the brain. The interneuron is then going to synapse with an outgoing motor neuron. Notice that the motor neuron, its cell body is here in the anterior horn. Okay, do you all agree this is the anterior horn of the spinal cord? Remember, spinal cord, what reaches up toward the back is posterior. So these are the anterior horns. The cell body is completely within the anterior horn. Also note that this neuron is a multipolar neuron. We see lots of dendrites, one axon. And that one axon is leaving out the anterior root. It's going to recombine with the nerve, and it's going to travel to the muscle. Okay, so this is a somatic motor neuron because it is going to be moving voluntary muscle. It's moving the biceps brachii. So that muscle is the effector, which is going to be moved because you voluntarily chose to move this muscle, and that signal was sent through this motor neuron or this efferent transmission. So that is an example of how the peripheral nervous system and the central nervous system work together, and it ties together the sensory neurons, the interneurons, and the motor neurons. Let's take a look at a nerve. So recall that a nerve is a bundle of axons, and another term used to describe an axon is a nerve fiber. So you can say that a nerve is a bundle of nerve fibers, or just a bundle of fibers, or it's a bundle of axons. On the top view, this is a highly magnified view of a nerve. The nerve would be, as big as your entire screen. And so imagine that the nerve is, I'm sort of pointing something bigger and rounder outside. And within the nerve, you know that there's going to be bundles of fibers. That bundle of fibers is called a fascicle. So I'm circling around a fascicle right now. And that fascicle would have around it a layer of connective tissue referred to as the perineurium. It's labeled here, the perineurium. And then within the bundle of neurons is going to be individual fibers. So here we're seeing individual axons. Notice that there are different sizes. Some of these axons are fatter, some of them are skinnier. And these have beautiful white around them. And that white, is myelin, made by Schwann cells. Now we know this is Schwann cells because this is a nerve, and nerves are only found in the peripheral nervous system, and Schwann cells are only found in the peripheral nervous system. We see blood vessels, right? These large vessels, evidence that these layers of connective tissue, like the perineurium, provide a space for blood vessels to travel along and within the nerve. Remember, Schwann cells are living cells. They need oxygen, they need nutrients, and so they need to have a supply of blood and nutrients and hormone signals just like any other cell in the body. And then each and every axon is also surrounded by a layer called the endoneurium. Not the most obvious thing here, just know though that it's surrounding each and every axon, I should say, and that the endoneurium is outside of the Schwann cell sheath, outside of the layer of myelin. So that would be a cross section of a nerve, you're looking at that. Oh, also, if we take a look, let's look around. Let's find a axon that does not have any white around it. And if I look around in here, I see, here's one, right here. Here is I can see there's an axon here, but I don't see any white rim around it. And you tell me, why don't we see any white? I see maybe two of them here without any white around it. Here's another one, right? I see an axon with no white around it. And if you just thought to yourself, oh, that's where a node of Ranvier is, you are correct. Remember, the nodes of Ranvier are the spaces in between the Schwann cells where there wouldn't be any myelin present. Now, if we take a look at the lower view, this is a much, not as high a magnification. The top one is a scanning electron micrograph. The bottom is a light micrograph. And here, the axons are traveling left to right. And you sort of get your eye trained to see these little places of cleavage. So here you see the indentation. That is a node of Ranvier. Here's another one. Here's a node of Ranvier. Another one. Here's a node. Here's a node. Okay, so those are the nodes of Ranvier. If you were to slice right at the node, there would not be any myelin present at that very minuscule spot, okay, along that axon. So what you're looking at here is just a bunch of axons traveling left to right, and you can see sort of this white shadow around the axons, and clearly you can see those nodes of Ranvier in 4 or 5 very easy to spot places. So that would be another nice view of a nerve to understand what a nerve is. This is one of those slides that I think is really, really important to your overall understanding. If this slide makes sense to you, then you're doing great in understanding so much of what has been taught to you so far this semester and in this unit. So let's take a look. Now, let me also say that I will not have you label this diagram, but I want you to spend some quality time looking at this diagram with me. So we recognize that we have the spinal cord. And coming up the spinal cord, we have 2 spinal nerves. So remember, the nerve is actually this bundle here, and it is the combination of the posterior root and the anterior root. Within spinal nerves, so you know that you have 31 pairs of spinal nerves. There's those 8 cervical nerves, 12 thoracic nerves, 5 lumbar, and 5 sacral nerves, plus the coccygeal nerve. Those 31 pairs of nerves all carry fibers, axons, that are part of all 4 of those divisions that I introduced to you at the beginning of this session. So let's take a look at what I mean by that. Within the spinal nerve, there are going to be somatic sensory neurons, visceral sensory neurons, autonomic motor neurons, and somatic motor neurons. Remember, these are those 4 divisions of the nervous system that I introduced in that box diagram at the beginning. So the somatic sensory neurons are shown sort of in the bright blue, and the visceral sensory neuron signals are in the dark blue. Notice that both of these blue neurons come in and travel toward the spinal cord by going up the posterior root. Notice here that we have the posterior root ganglion. You know that a ganglion is a cluster of cell bodies. The posterior root ganglion is full of sensory neuron cell bodies, and there they are, those little pushpin looking structures. We continue on, and I think you'll agree, right, that the posterior root only carries sensory information. So the incoming posterior root only has sensory information. The sensory information is going to come in, and it's going to synapse with interneurons that are themselves found within the gray matter of the spinal cord. Remember, the gray matter of the spinal cord contains cell bodies and dendrites. And we see that specifically those sensory neurons come in and synapse on interneurons within the posterior horn of the butterfly, of the spinal cord. Those interneurons are then going to travel a short distance, and they're now going to synapse with motor neurons, two different flavors. You have the autonomic motor neurons. These are going to be always in the lateral horn. And remember, the autonomic motor neurons are going to be very critical to the sympathetic nervous system. That'll be introduced to you in the final presentation of this unit. But the lateral horn is where you find those cell bodies for the autonomic motor neurons. And then the anterior horn is where you're going to find the cell bodies for the somatic motor neurons. So then the autonomic and somatic motor neurons, those fibers are going to leave out the anterior root. Everything traveling through the anterior root is motor function. All of those 4 fiber types now merge together and travel throughout the body as a nerve. So let's think about really quick, what kind of information is traveling into the spinal cord from a somatic sensory neuron? And go back and look. That's the kind of information coming from your skin, from your muscles, from your joints, and from your special senses. And then visceral sensory information is coming in from your gut, from your lungs, from your stomach, from your liver. All those viscera are also sending sensory information in. Leaving the yellow neurons, the autonomic motor neurons, those signals are heading to involuntary things such as cardiac muscle, smooth muscle, glands. And then the red signals would be leaving and traveling to your voluntary skeletal muscles. So hopefully that pulls together more of this story. Now, on the right-hand side of this diagram, the author has simply colored those 4 regions of the horns. Now I'm not going to ask you to memorize this, but I do want you to know that the sensory regions are in the posterior horn, the autonomic is in the lateral horn, and the somatic is in the anterior horn. And one more term while we're here, and that is, you see a bracket here. These blue regions are the sensory nuclei. Remember that a nucleus is a cluster of cell bodies in the CNS. And recall, what do we have here, right? In these horns, we see clusters of cell bodies. So another way of describing the horn would be to say these are the somatic nuclei. The lateral horn is essentially the autonomic motor nuclei, and nucleus, and the red region would be the anterior horn, which you could also refer to as the somatic motor nucleus. We have seen vertebra and we have seen the spinal cord. We have not really seen it together. So for the first time, we're getting a superior view. We're looking at a vertebra. And tell me, what kind of vertebra is this? And if you said thoracic, you're right. And the only reason I can see that right here is that I can see that there is a little divot right here for where the ribs would articulate, and this is where the costal cartilage would be, where the articulation occurs. So because I'm seeing that, and I'm also seeing no transverse foramina, this is a thoracic vertebra. We see the spinous process here, and we get a sense now that the spinal cord is only taking up maybe 60% of the diameter of that vertebral foramen, that opening within the vertebra. And we can clearly see the gray and white matter regions of the spinal cord. This diagram also gives us a quick reminder, a quick review of the meninges. So you can see here that there is the epidural space. That's this fat-filled space that I'm pointing to right here, also up here. And that is the region that nurse anesthetists and anesthesiologists, when they're giving an epidural, they inject that anesthetic right into this fat layer, right? You don't want to have that needle go into the spinal cord proper. Now right below that is the dura, that's that hard layer. Then there is the arachnoid motor, and you can see those little spider web-like structures. In that dark region, that is the subarachnoid space. The subarachnoid space, recall, is where you have cerebrospinal fluid. And then shrink wrapped to the surface of the spinal cord itself is the pia mater. So those aren't labeled, but a good reminder of those meningeal layers. Here, for the first time, again, we're seeing the spinal cord interacting with the vertebra, and we see the spinal nerve protruding out and coming through the opening. Remember, this would be the intervertebral foramen. That's the space between the vertebra that would allow for the spinal cord to protrude in or out. And out here we see the entire nerve, right? This circle represents the entire nerve. In this side, we can see the posterior root ganglion, right? That cluster. If we were to cut into that ganglion, we would see a bunch of sensory cell bodies, and they would be unipolar in their shape. So that is the posterior root. Here is the anterior root. When they come together, that is the nerve. Now, as soon as we're outside, as soon as that nerve protrudes through the intervertebral foramen, we see that the nerve begins to branch. And there's a posterior branch and an anterior branch. Now, the fancy word for those branches is a ramus. So we have a posterior ramus and an anterior ramus. The posterior ramus is relatively small, and it's simply going to be going to the muscles of the back. So this posterior ramus is only going to carry motor information back to the back muscles, maybe to some virorector spinae muscles along the vertebra, and it's also going to be receiving sensory information from your back. So if you can feel along your back, then this nerve would send that sensory information in. The larger and far more substantial branch is the anterior branch. There's a lot more in front of the spinal cord, if you will, than there is behind. And so the anterior ramus is going to reach around and come around. Remember, this is a thoracic vertebra. So there would be a rib here, and this anterior ramus you could also refer to as a thoracic nerve, right? It's one of those intercostal or thoracic nerves that's coming off from the spinal cord. Right here, this little teardrop or ear ring, this is a sympathetic trunk ganglion. We're going to see those sympathetic trunk ganglion in the next presentation when you take a look at the sympathetic nervous system, and you'll see that there's a series of these ganglia that travel up and down along the spinal column called the paravertebral ganglia, right next to the cell bodies in the vertebra. So here's a word that I think I mentioned in passing, but I want to give you a better description of it, and that is a dermatome. Each of your spinal nerves, except for C1, is responsible for the feeling of a region of your body. And you'll see this map in a moment. And so each region of your body is broken up into a dermatome map. Derm, right? Dermatology, or the dermis, right? So the dermatome map is a map of your skin regions, and each dermatome, like I said, is responsible for the feeling of just a small region of your body surface. You also, in the next presentation, will hear a little bit about referred pain, and I'll introduce that here to you as well. But if you are having appendicitis, for example, the appendix internally is innervated by spinal nerve T10, but also your umbilical region on the surface is also innervated by T10. So when someone's having appendicitis, they feel pain externally to that region that is also served by the same nerve. So let me show you that picture here in a moment. So here is a dermatome map, and you're not going to memorize all of the detail, but there are a few very important key facts that I want you to appreciate about this. So you can see that all of the nerves, except for C1, you don't see C1 anywhere on this diagram. C1 does not have an associated dermatome. Also, the front of your face, the feeling of that is controlled by cranial nerve number 5. But the rest of your body is broken up into these regions called dermatomes. So we see C2, right, kind of back by your ears, C3, up kind of where your hyoid bone is, right, C4, kind of heading around along the clavicle, C5, and then we see C5 continue down the arm, C6, 7, and 8 down here by the digits. Then the thoracic region, what kind of where we would expect it to be, it is along your thoracic and abdominal region. Lumbar region is essentially down the front of your leg. Sacral region is essentially down the back of your leg. Okay, so the lumbar region is down the front of the leg, anterior leg and thigh, and the sacral region is primarily your buttocks as well as your posterior thigh and leg. So this will have an importance. So for example, if you are having a appendicitis, like I just mentioned, T10, if you're having appendicitis, you're going to feel pain right through this region externally, almost like it's coming from the surface of the body. The actual organ that's in trouble is the appendix internally, and that nerve that serves the appendix is also T10, and that's where there's confusion. Basically, there's an SOS signal being sent out by your appendix, right, saying, I'm in trouble, I'm inflamed. But the brain can't quite figure out, is that signal coming from a somatic sensory neuron or a visceral sensory neuron? And so that's where we get this referred pain. Let's make sure we understand the significance of that dermatome map when it comes to spinal cord injuries. So spinal cord injuries, right, they're devastating. And part of the reason they're devastating, as we know, neurons are unable to regenerate. So once a neuron is cut off, or once a neuron is destroyed, or once a spinal cord or part of the brain is destroyed, there aren't other cells that can come into the rescue. There aren't stem cells that can repair that damage. Recall too that neurons do not have centrioles. So even if they wanted to divide, without centrioles, these cells are unable to do mitosis. They can't divide and make more cells like themselves. So let's take a look at different types of paralysis from spinal cord injuries. If you have a spinal cord injury at C4, that's pretty high up in the neck, right? A high cervical spinal cord injury. And when I say spinal cord injury here, I'm referring to a bilateral complete break. That individual is going to be paralyzed, everything in red. So from their neck down, they're not only going to not be able to feel, but they're also not going to have any control of the muscles and body functions from that region down. Pretty devastating. C6 injury, very similar. You see they have a little bit more movement up around their neck, but for the most part, they still are a quadriplegic. They've lost the use of all 4 limbs. Now, if the person's injury is around T6, thoracic 6, notice now that only their abdominal region and legs are paralyzed, that they have full use of their arms. As a result, these individuals are referred to as paraplegics. They have use of their arms, but their legs are not. And then if you see someone with an L1 injury, then they would have full use of their arms, full use of the thoracic and abdominal region, but they would also, remember that the lumbar and sacral nerves go down the front and back of the leg, so they would not have use of their legs. Now, if you see someone in a wheelchair and they're paraplegic, and you know that because they are moving themselves, right? They're using their arms to push themselves. You can tell a difference between a T6 and an L1 paraplegic basically by looking at their core strength. So if you see a person sitting up very strong, and they have very good core strength, then you know that their injury is down around L1. If instead they seem to be slouching and they have poor core support, then you can suspect that their injury is more in the T6 range. Now the line here basically is at the line here is basically at around T1. So if anything's above T1, then that person's going to be a quadriplegic. Anything down in the rest of the thoracic or lumbar region, that person's going to be a paraplegic. So let's go back and just take a look at that. So again, if they have the break at, if you see, look at this now, right? So if they have a cervical break, they're going to lose all that, they're going to lose wherever the point of break is from that point down. So you see cervical. So cervical is largely taking out their arm. So if they have something above in the C8 or T1 range, it is going to really wipe out their arms. Anything below that, they're going to have full use of their arms. In addition to quadriplegia and paraplegia, another type of paralysis is hemiplegia. So let me describe these three to you. In this particular diagram, the lighter colored gray area is the area of paralysis. So in a quadriplegic, again, something in the cervical region or T1 and above, you lose all four limbs. If you have a break in the thoracic or lumbar region, T2 down, you're going to be a paraplegic, and you're going to lose portion perhaps of your abdominal region and your legs. But there's also hemiplegia, and hemiplegia is basically paralysis on one half of the body. This is 99% of the time going to be damage to the brain. So you know that if there is damage to the right side of the brain, that it's going to affect structures and functions on the left side of the body. More specifically, if a person has brain damage or stroke that affects their right frontal lobe, right? Remember, the frontal lobe is your motor lobe. So if a person affects their right frontal lobe, then they're going to see paralysis of some extent on the left side of the body. That would be a type of hemiplegia. So if you've seen someone or know someone who's had a stroke that's affected their movement, you'll notice that the paralysis is only on one side of the body. So they have some form of hemiplegia. So know your three types of paralysis. Let me just go back and make sure that the plexuses make sense to you. There are 4 plexuses, recall. These are the groups of nerves that come off the spinal cord. The most superior of the plexuses or the plexi is the cervical plexus. The cervical plexus is primarily made up of nerves C1 through C4. Of this region, there's only one nerve that you were asked in lab to know, and the nerves you know in lab are the same nerves you need to learn here for the lecture portion of the exam, and that was the phrenic nerve. The phrenic nerve. And the phrenic nerve is so important because it goes down to the diaphragm to control breathing. Now, there's a phrase here, and this will make sense in a minute. C3, C4, C5 keep the diaphragm alive. C3, C4, C5 keep the diaphragm alive, and here's why. If you take a look, let's take a look. Here's the phrenic nerve. Now, I would never ask you to label the phrenic nerve amongst all this mess, but it's the only nerve that you've been asked to know that is derived from the cervical region. So if there's any nerve up in the cervical region, it must be the phrenic nerve that I'm asking you to identify. So if we take a look at the phrenic nerve, we see that the phrenic nerve is derived from a little bit of C5, a little bit of C4, and a little bit of C3. Right? So portions of C3, C4, and C5 all come out to form the phrenic nerve. This phrenic nerve then travels down across the thoracic cavity and inserts into the diaphragm, controls your breathing. So what's the indication of this? Why do we care? So if you have a break at C5, if if you have a break, let's say a break at C5 or C6, right, spinal cord injury, you are going to be a quadriplegic. But because the break was below C3, C4, C5, the phrenic nerve did not get affected, and you will be able to breathe independently. However, if you have a break very, very high, let's say at C3, so if you have a break at C3, right, that means that C4 and C5 are cut off. The majority of your phrenic nerve is coming from C4. If you have a break at C3, you will not be respiratory independent. You will need to be on a respirator. So not only are you a quadriplegic, but you also are unable to breathe without a respirator assisting you. So that's a huge deal, right? For cervical breaks, is the diaphragm intact? Is the nerves, are the nerve signals going to the diaphragm intact? Let's take a look at the brachial plexus. This one's easy. You know that these nerves are traveling to the arm, to the brachium, hence its name, to supply the muscles of the arm. The brachial plexus is made up of spinal nerves C5 through T1, C5 through T1. Okay, and there are 5 nerves that come off from this, these nerves, this plexus, that go to the arm. You had to learn all of these in lab. You're not going to have to label them in this sort of arrangement, but recall there was the musculocutaneous nerve, median nerve, axillary nerve, radial nerve, and ulnar nerve. Those are the 5 nerves you need to know that run down the arm. And of significance was that the musculocutaneous nerve, remember, is the nerve that serves your biceps brachii. So if you had damage to the musculocutaneous nerve, or if you had damage up here to C5 and C6, right, you would have lack of control of your biceps brachii. Again, remember, these cervical nerves are critical to the arm function. And so remember, we said if a break is at T1 or above, you're going to lose the majority of your arm function. And just take a look at the lumbar plexus. That is L1 through L4. These nerves are going to travel down the front of the leg, right down the front of the thigh, and from this plexus, the nerves that the 3 nerves that you had to learn that were most important were the lateral femoral cutaneous nerve, and that nerve is easy, guys. It's lateral. It travels down the lateral thigh along the femur near the skin, right? Lateral femoral cutaneous nerve. There's also the femoral nerve. This nerve is critical because it goes down to the quadriceps femoris group. So the femoral nerve travels right down the front center of the thigh and goes to your quads. And then the obturator nerve travels more medially, and this is the nerve that travels through the obturator foramen, right? You can see the obturator foramen up here. And this nerve travels more medially. This nerve goes, as you're learning in lab, to the gracilis and to the adductor muscles. So let's just kind of take another look at this, not to memorize this information, but just to finally maybe put this all together. So this diagram is about the femoral nerve. The femoral nerve is coming from branches of L2, L3, and L4. L2, L3, and L4 come off the lumbar plexus. Parts of those go down to form the femoral nerve. As you see, it comes straight down the front of the thigh, and it goes to muscles that you know. It goes to the rectus femoris, the vastus, right? It goes to your 4 quadriceps. Oh, by the way, it also goes to your sartorius, your pectineus, your psoas major, and your iliacus, right? So pretty important. So all of those muscles that we've learned about in the thigh. If you lost function to or had damage to L2, L3, or L4, you would affect this nerve's function, and you would lose some or all control of these muscles. You would also, those same L2, L3, L4 would be responsible for the feeling here in the leg. So this green represents the dermatomes that are part of the femoral nerve. The sacral plexus, our last one, goes L4 to S4. Notice it does overlap with the lumbar plexus. The most significant nerves here, notice that they L4, L5 come through the intervertebral foramina, then S1, those nerve roots come through the dorsal sacral foramina of the sacrum. Most significant here is the sciatic nerve. The sciatic nerve is really a double nerve. It's really the common fibular nerve wrapped up with the tibial nerve. And these two nerves travel down. They split, as you know, down around the popliteal region. And they are important because it goes to the hamstring group, right? So the sciatic nerve travels to the hamstring group, and then later you'll see that the gastrocnemius is also innervated, and the tibialis anterior also innervated by portions of this nerve. So that is just a review of the plexuses, and I hope now that you're beginning to understand why in Lab 6 you were asking to know the spinal nerves that were controlling so many of the major muscles, of your body. And there's quite a few of those that you're having to learn. You also know about the cranial nerves that control the trapezius and the sternocleidomastoid. So let's take a look at a reflex. Reflexes, there's a presentation posted after this that goes into reflexes a little bit. So this will basically help you prepare for that. But a reflex, we can divide up into 5 parts. Right, a reflex, here's a piece of skin. And let's say this is in your finger. Let's just say it's in your finger, okay? So this pin has poked your finger. As a result, sensory neurons in your skin, right, are going to travel. Now this would be a somatic sensory neuron, right? Same colors we saw before. Somatic sensory neuron. It's going to come in, go to the posterior root. There is the cell body for that sensory neuron. And it's going to come in. It's going to synapse with an interneuron in purple. And then synapse with a motor neuron heading out the anterior root, going out the motor side and going, to this case, it looks like the biceps brachii. So it looks like you maybe punch your finger, and as a result you kind of pulled your hand away, and your biceps brachii was a part of that activity. So these are the 5 steps, right? There is a stimulus, there is a nerve impulse, there is the nerve impulse being integrated by the spinal cord interneurons, a synapse going out, a motor neuron, to the number 5, the effector, the muscle that is responding to that. Now this is really the only example where your voluntary skeletal muscles are not voluntary. I mean, when you are reflexively pulling away from pain, or burning your finger on the stove, or stubbing your toe on a chair, you are not voluntarily, right? You're not making the decision to move your hand or redistribute your weight or move away from the pain. That's happening for you at the level of your spinal cord. And that's what a reflex is, an automatic response to minimize pain or minimize damage, right? And you pull away even before you were even consciously aware of the pain or the cellular damage. Now, reflexes can be described in a couple different ways. If the reflex is on the same side of the body as the effect, so in other words, if you touch a hot stove with your right hand and you pull away from the hot stove with your right arm, that's all happened on the same side of the body. So if the receptor, right, the burn and the effector muscles that moved away are on the same side of the spinal cord, we refer to that as an ipsilateral reflex. Remember, we learned back in Lab 1, ipsilateral means on the same side. If instead you stub your right toe, and in stubbing your right toe, you lift your right foot away from the pain, right, and you also extend your left leg in order to catch your body weight, it happened on the other side of the spinal cord, right? There was definitely information that crossed over to the other side of the spinal cord in that reflexive move. So that's referred to as a contralateral reflex. Let's take a look at these. So here, both of these would be considered ipsilateral because they're happening on the same side. So for example, you burn your hand, and you pull your hand away, right? It's happening on the same side of the body. That's ipsilateral. There's another term that can be used to describe reflexes, and that is monosynaptic or polysynaptic. So most of your reflexes are polysynaptic, and all that means is this. So as the sensory neuron sends that signal in, again through the posterior root, comes in, goes to an interneuron. There's one synapse. Then the interneuron speaks to a motor neuron. There's another synapse. There's two synapses. Because there are more than one synapse, we call it a polysynaptic reflex. This is the way most of your reflexes are wired. However, there is a reflex, and we've all experienced it. It's sort of the classic example of this, where there's only one synapse involved. So there's monosynaptic. And the example of this is your quadriceps femoris reflex. We've all had a reflex hammer tapped on our patellar tendon. As a result, our quadriceps muscles send a signal. That signal is brought into the spinal cord. This is in the lumbar region, right? This is the quadriceps muscle. We know the front of the thigh is a lumbar region nerve. So it comes into a lumbar nerve, and only synapses with one outgoing motor neuron, one synapse. And that motor neuron then comes out and causes the quads to contract. When the quadriceps contract, you know that you extend your leg out at the knee. So this is a monosynaptic. It's also ipsilateral. It's on the same side of the spinal cord, but it's also referred to as a monosynaptic reflex because there's only one synapse. Now, this is not your typical wiring, and this is relatively uncommon. So the patellar knee reflex is the classic example of a monosynaptic reflex. Just a couple more things about reflexes. The patellar knee jerk reflex is a stretch reflex. What's that mean? Think about it for a second. You've had this test done on you. So you walked in the doctor's office, you're sitting up on the bench, your knee is hanging down, and the doctor just gently taps the region just below your patella. Now you know what's there. You know that that's where that tendon is, down to the quadriceps that attaches to the tibial tuberosity. So they tap in between the patella and the tibial tuberosity. In doing so, they're stretching that tendon. Well, when the muscle is stretched, the muscle says to itself, remember this is a skeletal muscle. The muscle says to itself, I just got stretched, but I don't remember asking or being told to stretch it. Sort of as an unexpected response, right? Or a stimulus. So as a result, when the muscle gets stretched, the muscle contracts, right? So the response to that tapping is for the muscle to contract. That contraction is what causes the extension at the knee. Okay, so that's a stretch reflex. And this is evidence that our muscles not only receive motor signals, but our muscles also send out sensory signals. If I were to say to you, right, what kind of neurons are in our muscles, you would probably say motor, because we know that our muscles respond to neurons, to motor neurons that come from our spinal cord and tell our muscles to move. But your nerves also have sensory neurons in them. And so these sensory neurons are reporting to how much your muscle is stretched, right? So if your muscles get stretched, then that signal is sent to your spinal cord as well. So your muscles have both motor and sensory signals going to them. One final reflex to describe, and it's a really cool one, was described by the same dude who was famously named for the Golgi apparatus, right? Remember the Golgi apparatus, one of the cell organelles. The same dude was named for describing this Golgi tendon reflex. You know that tendons connect muscles to bones. And you know that tendons are made up of dense, regular connective tissue mostly collagen, very, very strong, but they do tear, right? We know that people have their tendons torn from excessive stretching or some sort of trauma. So Golgi tendon organs are basically the nerve endings located within the tendon, close to where the muscle and tendon join. If you've ever watched a bodybuilder going for a new weight or a really heavy weight, and they're all psyched out, and they go to lift that heavy weight, and it's almost like their body turns to jello, right? They're trying with all their mind to lift that weight, but their body kind of goes limp. Here's what's going on. This nerve ending, right, that is found, here's the Golgi tendon organ, if you will, and this is the nerve ending. And if this muscle is being strained to the point where the tendon is being stretched and the tendon might break off from the bone, right? It's so much force. These sensory neurons send a signal to the spinal cord, and basically it causes this muscle to go limp. So in the case of most reflexes, right, we think of a muscle contracting. Muscle responds to move away. But in the case of the Golgi tendon reflex, the reflex is actually for the muscle to go limp, for it to relax. And the person, of course, drops a weight, unable to contract that muscle. There are a whole bunch of reflexes. The only one you need to know is that patellar knee-jerk reflex. But recall, the patellar knee-jerk reflex is testing your quadriceps muscles. That is your femoral nerve. Your femoral nerve is derived from L2, L3, and L4. So when the doctor is testing your patellar knee-jerk reflex, they're really making sure that your lumbar nerves are intact. There's a whole bunch of other tests, right? From biceps, triceps, abdominal reflexes. There's a Cremaster reflex that can be done in males. There's an Achilles reflex. There's a plantar reflex. And all of these reflexes tell the doctor if your spinal cord is intact. Now, normally we don't do all of these reflexes unless there's some concern. So if you just go in for a normal physical, the doctor's going to ask, have you had any issues with motor control, coordination? And if you report negatively, they're not going to dive in any more deeply. But if you are complaining about some sort of clumsiness, or you've had an accident and you're in the ED, in the emergency department, then they're going to do a more thorough examination of your reflexes to assure that there's no spinal cord injury. Let's review a few structures of the brain while we're here with this diagram. And you already know about the lobes. You already know a couple of key structures. So let me just remind them to you. So you have the central sulcus. The central sulcus is this groove in between the frontal and parietal lobe. Remember, this is where we have the motor strip or the pre-central gyrus in the frontal lobe. And then on the other side, we have the post-central gyrus or the sensory strip. Wernicke's area is this area that kind of is right here, sort of on the border of the parietal lobe and the temporal lobe. Wernicke's area, recall, is a speech area responsible for receiving and perceiving and understanding language. And then the other speech area is over here. It's called the Broca's area, shown here in purple. The Broca's area is in the frontal lobe. Notice that it's next to the motor strip, and it is about motor speech issues. So any damage here to the brain would result in a person unable to coordinate the muscle movements necessary to get speech out, motor movement or motor area of speech. This diagram also reminds us of the primary region for the different senses. So the temporal lobe, remember, has the auditory cortex, the primary auditory cortex. The occipital lobe has the primary visual cortex. The parietal lobe is the primary somatosensory cortex. So if you could feel something on your skin, one of your dermatomes, that information is coming up to your sensory strip. And the frontal lobe has the primary motor cortex. Specifically, that motor strip is the region that's going to send signals to your body to move your skeletal muscles. Your primary gustatory cortex is in the insula. Remember, the insula is so insulated that you can't see it. You're only going to see the insula if you were to pull up on the lateral sulcus, pull down, and hiding back there is another lobe called the insula, on which you have the primary gustatory cortex. You're looking here at the left lobe. Remember that Wernicke's and Broca's area are, for the vast majority of individuals, on only the left side, that the left and right brain are not mirror images of each other. Now let's take a look at this blue strip of land. So this is the precentral gyrus, also called the motor strip. And if I were just to take out that strip of brain matter, imagine this is like a birthday cake, and I'm going to slice that piece out. Okay, I'm going to make a slice here in this brain cake, and then I'm going to lay this blue strip down on the plate. I would get something like this. So let me help you get your orientation. So we sliced that blue strip out, that precentral gyrus, that motor strip. Let's get our bearings. So this is only half. This would be one half of the brain. The other half would be over here. It's not shown. This deep groove here would represent the longitudinal fissure. This would represent the ventricle, or you could think this is sort of like the corpus callosum that connects the two hemispheres together. Here, this is the lateral sulcus, that groove on the side. So that means that everything above this is going to be frontal lobe. This is going to be temporal lobe, where my pointer is. And as I just showed you, if you go deep to that lateral sulcus, you run into the insula. So right here, where my pointer is, that's where the insula is. And what we notice is that the artist has drawn this strange little man over the top of the motor strip. This is called the Homunculus Man. And basically, the bigger he is, what that represents are the number of neurons that leave from this strip and go to that part of the body to control movement. And we see that, about a fourth or so of this motor strip is related to the movement of muscles on the face, right? There's a lot of complex movement for swallowing, for chewing, for jaw movement, for vocalization, for your lip movement. Speech is the most complex motor thing that you and I do, right? I mean, when we talk, think about what's going on. How quickly your tongue and your lips and your vocal cords and all of those, all those structures in your throat are quickly changing in order for you to speak. It's an amazing process. So a lot of muscles involved with speaking and moving the face and the throat. Then we know that our hands are very, very important for movement, a lot of fine motor movement. So the hand has a large amount of this strip, and then the rest of the body has the rest of it, right? So we see this region of the precentral gyrus. Now what this means, I'll show you in a moment, but this also helps you to predict where brain damage is. If a person has damage to the motor strip, they're going to have paralysis related to this region, but remember to be on the other side of the brain, other side of the body. So here is a map. We're seeing basically the same picture, a different artist. Here is the motor strip. We're in the frontal lobe. Here it is. If we go just one strip back, that is the primary somatic strip, or the sensory strip, or the post-central gyrus. We see the same idea. We see the same dude kind of stretched out. Here, though, this is referring to the parts of the body that send sensory information, so surface somatosensory information. So you can see your legs and feet, right? Hand, also very sensitive area. Face is extremely sensitive. So we see, again, a large part of the strip dedicated to the face and to the hands. So let's make sure that this makes sense to us. So let's say this is the left motor strip. Left motor strip. And if I have brain damage to my left motor strip, and I have it right about here on the side of the brain. So imagine where this is, right? Sort of right about here where I'm pointing. Okay. So I've got damage. I've had a stroke, some sort of brain damage or a traumatic brain injury, and it has affected this part of the brain. I do an MRI, and it shows that this part of the brain is dead. Well, even before I do the MRI, I'm kind of guessing this is where the damage is going to be, because what am I going to notice? I'm going to notice this is the left side of the brain. I'm going to see paralysis on this person's body, and their arm and their leg is going to be paralyzed, right? Sorry, their arm and their hand. So their arm and their hand is going to be paralyzed. I'm going to know, right, almost exactly where on the brain that damage is because of the symptoms. If instead that damage is down more laterally, then I'm going to see more issues with speech and facial paralysis. Okay, so you get the idea that wherever this damage is on the motor strip is going to affect movement on the opposite side of the body. Likewise with the sensory strip. So if there's damage to the parietal lobe that affects a region of the sensory strip, that person's going to have difficulty sensing vibration and touch in those regions, again, contralaterally. I love this dissection. On the left, we're actually looking at a dissected brain, and you can see all of these nerve fibers coming up. Ooh, I just said something wrong. I said nerves, right? There are no nerves here. This is in the CNS, so these are tracts. So we're seeing all these tracts coming up. Remember, a tract is a bundle of axons within the central nervous system. And we see that these tracts are white, right? Because they're covered in myelin. And remember that this myelin is produced by oligodendrocytes, the glial cells that make myelin in the CNS. And we see too that most all of the signals that come up from the spinal cord into the brain pass right through this region. See where I'm drawing? And you know that this is the diencephalon. Specifically, this is the thalamus. And the thalamus is oftentimes called the gateway to the cerebrum, because almost everything that passes up through the spinal cord to the brain passes through the thalamus. This slide is a little messed up, but you also have this slide posted in the Cranial Nerve Review. So just let me remind you that you are learning your cranial nerves, numbers 1 through 12, and it's important that you know the numbering of the nerves, 1 through 12, the names of the nerves. You need to know if the nerve is a motor only, sensory only, or is a mixed nerve, does both. And you need to know the major, what that nerve is either sensing or what it's moving. So make sure that you're using these mnemonics. One set of mnemonics is to help you learn the name of the nerves, 1 through 12. The second set of mnemonics, for example, some say Mary Money, but my brother says big brains matter more. These mnemonics are to help you learn the function of the nerves, 1 through 12. So S is for sensory, sensory, motor, motor, both. So memorize one of those phrases, one each, if it helps you to keep these cranial nerves straight. There is a worksheet posted which helps you sort of work through the cranial nerves, and that worksheet also has the answers posted, and there will be a section on the exam very, very similar where I'll give you a story where you will need to identify the nerve involved with those senses or movements. The rest of, and the last little bit of this presentation is a little bit in advance if you haven't already watched the autonomic nervous system presentation, but that's okay. This will sort of act as an overall review. So let me just say that part of the exam is going to cover the autonomic nervous system, and there will be a lot of comparing and contrasting between the somatic nervous system, aka your voluntary nervous system, and the involuntary or the autonomic nervous system. So here we have the somatic nervous system, aka the voluntary nervous system. This is going to look just like you're used to. There is a sensory neuron in the skin. There's a sensory neuron that goes into a nerve, goes in the back door through the posterior root, comes into the spinal cord. Comes into the spinal cord where there's now going to be interaction with a motor neuron, and then out the anterior root to the effector, to the muscle. That's the same sort of wiring that we've seen in all other things related to the nervous system. As we get into the autonomic nervous system, what we're going to see now is that on the motor side, there are going to be two neurons. There is a preganglionic neuron. We see that here. That preganglionic neuron travels from the spinal cord down the anterior root and goes to a ganglion. At the ganglion, there is now a synapse. The synapse is to a postganglionic neuron that is going to travel from the ganglion to the effector, in this case, the lungs. So the sensory side is the same. We still see a sensory neuron going from a visceral, this is a visceral sensory signal going from the lung, coming up, going in the back door, there's the cell body, and then coming down, synapsing on a motor neuron, and then out. And what we see is a two neuron system, preganglionic and a postganglionic neuron. Again, there's going to be more details about that in the autonomic nervous system presentation. On the exam, there will be a Venn diagram. You know what a Venn diagram is. It's the two overlapping circles. And then I will give you a number of facts that you will then assign to either being the somatic nervous system or the autonomic nervous system. So for example, if I gave you a fact and I said voluntary control, right, you would assign that feature to the somatic nervous system. And if instead I said that it was an involuntary control, you would assign that to the autonomic nervous circle. If I said, however, that the effector was skeletal muscles, you would assign that to somatic nervous system. If I said that it went to cardiac muscle, smooth muscle, or glands, you would assign that feature to the autonomic nervous system. A couple of interesting things here, though. The somatic nervous system only excites. So when you send a signal from your brain, from your frontal lobe, specifically from your motor strip, and you tell your biceps brachii to contract, it sends an excitatory signal. It sends a signal that says turn on the muscle, and when that signal gets to the muscle contracts. But what you're going to see when you get to the autonomic nervous system is that the effects are not just excitatory, but they can also be inhibitory. So the autonomic nervous system is more like a dial. You're going to dial up or dial down, but you're not just flipping a switch on like you do when you contract your muscles. So read through these little facts. These facts will make sense in light of what we discussed in that presentation as well. And I'll use these facts to ask questions for a Venn diagram to assign features to the somatic nervous system, the autonomic nervous system, or to both. One more thing on the bottom here, axon properties. In the somatic nervous system, all of the axons are myelinated. They are thick. So recall when we look at a nerve, we see that the axons are not all the same size. Some are fat, thick, and some of them are skinny. Well, it turns out that the thicker ones are definitely going to be somatic nervous axons, right? Or I should say all the somatic nervous axons are going to be thick ones. So they're going to be thick and they're going to be coated with good old myelin. That thick axon actually allows the signal to travel faster and the myelin also helps to send that signal more quickly through the nerve. When it comes to the autonomic nervous system, the preganglionic fibers are going to be very thin, and the postganglionic fibers are going to be even thinner, and they're going to be unmyelinated. And recall that unmyelinated means that that the signal is going to move more slowly. I think that word may be below your screen, so just know right here, the postganglionic axons are thinner and they're unmyelinated, making them to have slower conduction, slower movement of the signal. In addition to a Venn diagram that compares and contrasts the somatic nervous system with the autonomic nervous system, I'll also have another Venn diagram on the exam to assign features of the parasympathetic versus the sympathetic divisions, and I'll use the facts from this table. So you know that, or you will know as you finish that last lecture, that the parasympathetic system is called the rest and digest versus the sympathetic, which is your fight and flight response. You're going to learn that the cranial sacral term is used to describe the parasympathetic, whereas the thoracolumbar term describes the location of the sympathetic division. You're going to learn that the parasympathetic system is not very divergent. Basically, or I should say, the sympathetic system is very divergent. What this means is extensive divergence means that when the sympathetic nervous system is activated, that everything gets activated together. Everything, your breathing rate, your heart rate, your pupil dilation, everything happens sort of simultaneously. Whereas in the parasympathetic nervous system, it is not as divergent, meaning that you could have one effect without another effect. The way I remember this is that it's possible to it's basically possible to digest your food or have an erection or go to the bathroom, right? All of those things are separate. So in the parasympathetic division, the rest and digest, those events can be separated from each other. You'll also see in that presentation that the parasympathetic system has long preganglionic and short postganglionic axons, whereas the sympathetic's reversed. You have short preganglionic and long postganglionic axons. And finally, just a couple more pictures of the sympathetic and parasympathetic nervous system. What you see here is that the parasympathetic, you have nerves coming off from both the cranium and the sacrum. Again, off the cranial region, you have cranial nerves 3, 7, 9, and 10. 3, 7, 9, and 10. And as I went into greater detail, you should know what those 4 cranial nerves are doing and how that effect is involved with parasympathetic responses. And then the sacral nerves also traveling over to the lower digestive region, bladder, and reproductive organs and their effects. Versus the sympathetic nervous system, where again you have very short preganglionic fibers traveling from the thoracolumbar region, only a short distance to the ganglion, and then a long postganglionic fiber that goes to the effector. So you should appreciate what the sympathetic and parasympathetic divisions do, their responses, as well as some of these anatomical details that are shown here in the diagram, but also shown in that chart. So I hope that this is a good sort of recap, pulls a lot of things together for you. And if you have any other questions, please let me know.